Hello everyone, welcome to our webcast today. I'm Cheryl Rogers, the Director of Marketing here at Golden Helix. Today our webcast is presented by Dr. Vivien Sheehan. She was our third prize winner in our second annual Abstract Challenge. Dr. Sheehan is an assistant professor at the Baylor College of Medicine, as well as a part of the Cancer and Hematology Center at Texas Children's Hospital, where she specializes in sickle cell disease. Today her presentation will focus on her research which suggests a potential treatment for sickle cell disease. Dr. Sheehan, the floor is yours.
Yeah, thank you so much. So yes, the focus of my talk is going to be on a gene I found to be a regulator of gamma-globin, which is a component of fetal hemoglobin, and its potential role as a therapeutic target in sickle cell anemia. And for this all to make sense, I'd like to first discuss sickle cell anemia a little bit. I am an MD PhD and take care of a significant number of sickle cell patients, generally the teenagers. And so this is a subject near and dear to my heart. I'm coming to it from a disease-based viewpoint rather than a genetics viewpoint or genomics viewpoint. So my goal is in the end to find additional treatments for our patients. So sickle cell disease affects about 100,000 Americans, and it affects millions more worldwide, especially in Africa, the Middle East, and India. Anywhere that's malaria endemic, there's been a selection pressure to preserve the sickle cell mutation. It's also the first disease that we knew the protein basis of, and the first disease that we knew the genetic basis of, that it's a single nucleotide change that leads to an amino acid change. In the beta-globin gene, instead of a hydrophilic glutamic acid, we have a hydrophobic valine. And so those hemoglobin molecules in the aqueous environment of the cell, especially when they're deoxygenated, try to bury that hydrophobic residue by making these polymers or rigid rods within the cell. And it changes the soft, flexible, discoid red cell into a rigid sickle-shaped cell, which is what gave the disease its name when it was first noted under a microscope in 1910, I believe, not by Herrick, but by his student. You can see the effect it has in the small vasculature. Instead of being able to deform and squeeze through the microcirculation, it instead leads to vaso-occlusion, which leads to hypoxic injury and then reperfusion injury. And since this is happening throughout the body, it affects many organ systems. So individuals with sickle cell anemia are at risk for stroke at quite a young age. 10% experience a stroke before the age of 11. They're at risk for pulmonary hypertension, acute chest syndrome, which is the #1 cause of mortality in our patients, nephropathy, priapism, which is an erection that won't go away, osteonecrosis, damage to the bone, especially of the femoral head. But what most people think about when they hear sickle cell anemia is frequent pain crises, which certainly greatly reduces the quality of life of our patients. And non-healing leg ulcers is another complication. Life expectancy is greatly reduced. It's 42 years for males and 47 years for females. And this is in the US and other developed countries where we screen for sickle cell and provide prophylactic penicillin, which greatly extends life. In low-resource countries where there's not even screening for this disease, there's a high mortality under the age of 5. So if we are able to increase the patient's fetal hemoglobin, which as you can see is a different gene from the beta-globin that contains the sickle mutation, we can help them make a different type of hemoglobin that doesn't sickle. And if there's enough in an individual cell, it's called an F cell, and it acts pretty much like a normal sickle cell, a normal non-sickle red cell, and can prevent many of these clinical complications. We all produce fetal hemoglobin as fetuses and as infants, but in normal individuals, the gamma-globin, which is the beta-like component of the 2-alpha chain, 2-beta-like chain hemoglobin, starts to decline soon after birth and becomes almost non-existent. Sickle cell patients still produce some gamma-globin, some fetal hemoglobin throughout their lives, but it's generally not enough to really modify their disease. So when we're trying to find a cure or disease-modifying agent for sickle cell disease, we typically focus on how to reactivate gamma-globin, which has proven to be quite challenging over the years due to a lack of understanding of how gamma-globin is regulated. How is it turned off? So this plot shows you the variability of baseline endogenous fetal hemoglobin in our study sample. It's been log transformed because the distribution of fetal hemoglobin is not normal. So in order to analyze it, we normalized it with log transformation. And we use this data, this baseline fetal hemoglobin data, as our phenotype. And we had whole exome sequencing on all of these patients. But I'd like to first discuss two different next generation sequencing methods and how they might give us different information. So in the late 90s, actually no, that was in 2008. In 2008, genome-wide association studies of baseline fetal hemoglobin as the phenotype were investigated and reported and found that BCL11A was a regulator of endogenous fetal hemoglobin. It was done with several thousand European patients and was then repeated in sickle cell patients. So people with variants in BCL11A had higher fetal hemoglobin levels. BCL11A was a negative regulator of gamma globin and fetal hemoglobin levels. However, BCL11A has a lot of functions in the cell and does not appear to be a good drug target. So it'd be very difficult to downregulate BCL11A with any other method than gene therapy. And even then, you'd have to target the erythroid-specific part of BCL11A. BCL11A variants were fairly common. About 20% of the population has them, which allowed them to be identified by genome-wide association studies, which detect common variants. And while this was a great advance in our understanding of how gamma globin was regulated, in the sickle cell community, it didn't appear to be a viable therapeutic target, and it also didn't explain all the variability of fetal hemoglobin levels. The variants account for less than half of the observed variability of fetal hemoglobin. So there's clearly more to be found. And so what we decided to do was whole exome sequencing to look at the rare variants that might contribute to the variability of fetal hemoglobin levels and might add to our understanding of gamma globin regulation. So whole exome sequencing identifies all the variants in the protein coding regions. It's able to identify rare variants that potentially might have a large effect that are causal. And it also had not been applied to this question of modifiers of endogenous or baseline fetal hemoglobin. And so our study population was 171 pediatric sickle cell patients, all with hemoglobin SS. There are different genotypes. These are the homozygous for the mutation I was describing earlier. Age 3 to 18 years of age. They were taken from 2 clinical trials, HUSTLE or hydroxyurea study of long-term effects, and SWITCH, stroke with transfusions changing to hydroxyurea. Russell Ware was the PI for both studies. And we did 2 separate analyses, and I'm new to statistics and genomics, and so how I approached the problem was I would first do my analysis using Golden Helix, and I had statistical backup. One of the authors on the title page, J.C. Crosby, is a statistician and is about to get her PhD, and checked my analyses. And especially for the linear regression, there was excellent agreement between what I did and what she did. So we did look at both analyses, linear regression, which is looking at the common single variant to phenotype relationship, so one SNP to fetal hemoglobin levels as a continuous variable. And then we also did a burden analysis, also sometimes called a gene-based analysis, and our cutoff was a minor allele frequency of less than 2%. We used a T2 test, and this is looking at the aggregate of all variants within the gene. And so it's a gene to phenotype analysis. And that'll be the analysis that I'll be talking about from now on because it seemed to produce the most interesting results. And perhaps that's not surprising given that this type of gene-based analysis can only be done with whole exome data or whole genome data, and so would not be identified by genome-wide association studies, even with the much larger numbers that were used to identify BCL11A. So our T2 burden analysis candidate genes are listed here in order of lowest p-value. And looking at our top 5, it jumped out at us that we had a little bit of a pathway already developing here. So AMPK, and that's the alpha-1 subunit, and FoxO3 are part of a pathway. AMPK activates FoxO3, and I'll show that in a schematic in a moment. And since FoxO3 has been implicated in erythroid maturation, this definitely highlighted those 2 genes for me, especially FoxO3. And in this box plot, you can see that individuals who did not have a FoxO3 variant had a higher fetal hemoglobin level than those that did. Nobody was homozygous for a FoxO3 variant, given its rarity. That's not surprising. And there were 2 variants that were found in 2 individuals, so 7 unique variants in 9 individuals. So FoxO3 also has many different functions. It's certainly important for oxidative stress, and in the setting of oxidative stress, reactive oxygen species, AMPK is upregulated and it activates FoxO3 through phosphorylation, as does SIRT1. In the presence of growth factors, like insulin, for example, AKT is upregulated, and it inactivates FoxO3 by phosphorylating it at 3 distinct serine residues. When FoxO3 is phosphorylated on those residues, it translocates to the cytoplasm where it's degraded. FoxO3 has also been identified as important for cell cycle control for apoptosis. It was found in a GWAS looking at longevity, which might be related to its ability to manage oxidative stress, and has a role in stress resistance. So this is a schematic of the FoxO3 protein showing our 7 variants. The ones in red were predicted to be damaging by polyphen 2 or SIFT. And I should have included a bean plot, which would show you that the variants that were predicted to be damaging were associated with lower fetal hemoglobin levels than those not predicted to be damaging. And you can see that they are scattered throughout the gene, bypassing those 3 phosphorylation sites that AKT phosphorylates to inactivate FoxO3. So we were encouraged enough with these results to start to pursue functional studies. Genomically, it looked like having a variant in FoxO3 led to a lower fetal hemoglobin level, so possibly if we knock down FoxO3, that would affect gamma-globin expression. So the first model system we used were K562 cells. They're often the first stop in investigating gamma-globin and globin switching. They are an erythroid leukemia cell line that are pretty easy to grow and express gamma-globin. The downside to them is that they don't express beta-globin. But in this system, we used siRNA to knock down FoxO3. And you can see in the Western blot here, scramble siRNA and control cells were used, and then FoxO3 specific siRNA. And knockdown of FoxO3 resulted in less gamma-globin production. And here's the RT qPCR data showing again that we achieved about an 80% knockdown of FoxO3 with the FoxO3 specific siRNA, and about a 60% knockdown of gamma-globin. We also then overexpress FoxO3 in this system, transfecting in a vector of either wild-type FoxO3 or the triple mutant FoxO3, which has those 3 serine residues that AKT uses to inactivate FoxO3 altered. And again, more FoxO3 led to more gamma-globin production. So the next step was to master a somewhat difficult laboratory technique of primary erythroid culture. So cancer cells like K562s are easy to grow, easy to transfect, easy to use. Much more challenging is primary cell culture. So primary erythroid cells can be obtained from cord blood or from the buffy coat of adult blood donors. And that was our source. We used the buffy coat of adult blood donors from our local blood bank and enriched the buffy coat for CD34 cells using Automax beads. The goal of primary erythroid culture is through cytokine and growth factor manipulation, have those CD34 cells become normal red cells in the end. Terminally differentiate into something that would look like it came out of the peripheral circulation of a patient. So we took the CD34 cells and expanded them, and during the expansion phase, infected them with the lentivirus that would knock down FoxO3. You then give the culture erythropoietin, which has been the kind of maturation arrest point. So now all the cells are maturing together as a group down the erythroid lineage. They're differentiating. And as the cells differentiate, their levels of gamma-globin decline and beta-globin rise. And so you can study the cells to the point where both beta-globin and gamma-globin are produced. And this is a great model system. It really mimics what's happening in your bone marrow right now. And of course, we'll analyze the cells by RT-PCR and Western blot to look at gamma and beta globin levels. So here again, we got about an 80% knockdown of FoxO3 with our shRNA lentivirus. And don't see my, oh, there it is. Okay. You can see that by RT-PCR. And here we have the gamma-globin levels. Actin was our housekeeping gene. Again, knockdown of FoxO3 led to reduction in gamma-globin, about a 60% reduction in gamma-globin. And we were very happy to see that beta-globin remained unchanged. Again, since our goal is to find a therapeutic for sickle cell anemia, we wanted a target that would be gamma-specific. So if it altered both globin chains, it wouldn't be a specific target. We certainly wouldn't want our sickle cell patients to be making more sickled beta globin. We want them to make gamma globin, fetal hemoglobin, and not sickle. So here's the Western blot showing knockdown of FoxO3, leading to reduction in gamma globin while beta globin remains unchanged. So this appeared to confirm that FoxO3 is a positive regulator of gamma globin. If its knockdown or mutation in an individual results in less fetal hemoglobin, less gamma globin, potentially its overexpression through drugs or other means would lead to more fetal hemoglobin without increasing the sickle beta globin. Unfortunately, FoxO3 levels can be increased with a few well-known drugs. Now, there's some debate about whether these drugs are acting through AMPK or through FoxO3. Different authors have different opinions on that, but in the end, as a therapeutic, it may not be terribly relevant since AMPK activates FoxO3 through phosphorylation. And also in our whole exome sequencing study, variants in AMPK were also associated with lower fetal hemoglobin levels. So metformin, fenformin, and resveratrol are three drugs that have been reported to increase either AMPK expression levels or FoxO3 levels. And so our proposed model for further investigation is that these agents may increase AMPK, leading to increase of FoxO3 and increase of gamma globin. And we've done some preliminary work on that, treating our primary erythroid cells with resveratrol, which increase gamma globin in a dose-dependent manner. And a possible mechanism for this increase in FoxO3 is that resveratrol causes FoxO3 to accumulate in the nucleus where it's not degraded. You can see that's in a dose-dependent manner, the laminin fraction being the nuclear fraction, the tubulin fraction of the cell being the cytoplasmic. This was done in K562 cells. And so some future directions for this project. The 171 patients that were subjected to whole exome sequencing are just a pilot. I've now collected 1,000 sickle cell patients, and they've all gone through whole exome sequencing at the Human Genome Sequencing Center at Baylor. I have not yet gotten my project level VCF to download into Golden Helix and start analyzing the data, but it should be available soon. And so if a promising target like FoxO3 can be identified with just 171 samples, there are likely more aspects of gamma globin regulation to be discovered with a significantly larger sample size. I'm collecting a validation cohort of 500 patients that will be used for more traditional sequencing validation like TACMAN. And so both linear association studies and burden analysis will be performed on these samples, with fetal hemoglobin as the phenotype as a continuous variable. And so this is another schematic of that idea, using our 1,000 as a discovery cohort, validate, and then verify by functional studies. And again, we have a great system in the K562 cells and then primary erythroid culture to further confirm, more accurately confirm the association. So our goals with this new analysis are to also find some additional FoxO3 variants that are associated with reduced fetal hemoglobin levels, further identifying what parts of the FoxO3 gene are important for this function. Use gene-based testing and pathway analysis to see if other genes in the FoxO3 pathway like AMPK and SIRT1 are associated with fetal hemoglobin levels, and also do genome-wide unbiased analysis to identify genes and variants outside of the FoxO3 pathway that segregate with fetal hemoglobin levels in this larger cohort. We really want to better understand how FoxO3 regulates gamma-globin expression, in addition to developing it as a drug target. We propose to use our primary erythroid cells to investigate where FoxO3 binds through ChIP-seq, through chromatin immunoprecipitation sequencing, whether it binds to some areas of the gamma-globin loci or other loci that regulate fetal hemoglobin, such as BCL11A, MYB, KLF1, doing this globally rather than looking at individual expression levels of these genes, taking a more unbiased approach rather than a candidate gene approach. We'll knock down FoxO3 in primary erythroid cells, and of course have as a control the same batch of Buffy code derived erythroid cells, and see by RNA-seq what genes are altered by FoxO3 knockdown, and assemble all of these different pieces of the puzzle using a gene set enrichment analysis to look at where there's overlap between the whole exome sequencing, ChIP-seq, and RNA-seq analyses. Again, given how many different functions FoxO3 has, it will be very helpful to look at this only in erythroid cells to get the erythroid-specific genes, and also look at it through several different modalities. So to conclude, our burden analysis of whole exome sequencing data identified 7 FoxO3 variants as associated with lower endogenous fetal hemoglobin in pediatric sickle cell patients. We expect to identify more variants in our larger whole exome sequencing cohort. In K562 cells and in primary erythroid cells, knockdown of FoxO3 reduced gamma-globin levels. Overexpression of FoxO3 increased gamma-globin. And this is us to conclude that FoxO3 is a positive regulator of gamma-globin and a viable drug target for the treatment of sickle cell and potentially other hemoglobinopathies like beta thalassemia. But certainly further work is needed to test that hypothesis and to elucidate the role of FoxO3 in gamma-globin regulation. So the functional studies were done by my postdoc, Yang-Kai Zhang, and all the sequencing was done by the Human Genome Sequencing Center on a collaborative view grant. Dr. Betty Pace at Georgia Regents University taught us how to do primary erythroid culture and has been a wonderful resource and mentor. The statistical analysis was confirmed by Dr. Eric Boerwinkle and his graduate student J.C. Crosby. The samples for the initial pilot study came from Russell Ware's clinical trials. So I appreciate everyone's attention, and I can now take questions.
Thank you so much, Vivien. I just have a couple of quick announcements, and then we will get to the questions. If you haven't had a chance, please feel free to enter your questions at this time in the questions pane. I will make sure that the webcast recording and slides are up either today, tomorrow at the latest. If you've attended, I will just email them directly to you, so look for those. Our next webcast is coming up on September 9th, and that will be presented by Hilal Al-Shakli of the Child and Research Institute at the University of British Columbia. He was our second place winner in our abstract challenge, and he will present his research surrounding novel genetic variants in a family with epilepsy. At this point, let's go ahead and take some questions.
Yeah, thank you. I see one from Daniel asking which are the most promising drugs to induce FoxO3 pathway activation. And of those three drugs, I'm most excited about metformin because it is a commonly used drug for type 2 diabetics and even pre-diabetics. It doesn't cause hypoglycemia. It's quite well tolerated, oral daily to twice daily dosing. And may not even need to be FDA approved since it is already FDA approved for other indications, and so could potentially be looked at in a phase 2 trial with some convincing in vitro data. Resveratrol is a very interesting drug, but and actually I did a clinicaltrials.gov search on it, and it's being used in close to 100 clinical trials. But what I've heard in the red cell community is that there are real concerns about its bioavailability. And of course, it is not FDA approved for other indications. Fenformin looks really good in culture, but that particular drug hasn't been used in patients other than possible oncological use for, I guess, about the last 20 years because of an increased risk of lactic acidosis. Metformin has a small risk for lactic acidosis, but it's significantly higher in fenformin. Okay, let's see. So yeah, another question from Suki is kind of similar. Any lead compounds used to treat sickle cell anemia now based on FoxO3 polymorphism studies? So yeah, I think metformin is the most promising, and something that will need to be established is how it plays with hydroxyurea. I didn't really get into that, but we do have a fetal hemoglobin inducing agent called hydroxyurea, which has several limitations. Not everyone responds to it. One of my genomic studies that was published in PLOS ONE recently was looking at variables that predict your response to hydroxyurea. That was actually kind of the initial reason for doing whole exome sequencing in this population. So not everyone responds to hydroxyurea with significant amounts of fetal hemoglobin. So could metformin be a booster to hydroxyurea? Is it better as a solo agent? These are questions that need to be answered. And again, primary erythroid culture is an excellent place to start. All right. Repercussions of long-term activation of FoxO3. I don't feel like there are negative repercussions to long-term activation of FoxO3. I think that its role in longevity, the numerous antioxidant therapies, of which resveratrol is one, suggests that increasing FoxO3 levels is likely to be helpful. I think there's no indication that having higher FoxO3 levels is harmful. I think it's likely to make someone better at managing oxidative stress, which is actually a big aspect of the pathophysiology of sickle cell. So I think it will be beneficial. How to set a control group. I'm not sure what that means, but perhaps the idea was in a clinical trial of some of these agents. Certainly you could have a randomized controlled trial with a placebo, or you can, in a phase 2 more type of model, you're comparing what their fetal hemoglobin was before therapy and then on therapy. Patients' fetal hemoglobin levels after they stabilize at around age 3 stay steady, and so it's not like it fluctuates from month to month. So if a patient has a fetal hemoglobin level of 10 at the start of the study, it's unlikely to vary even a percentage point significantly up and down. So any change could be attributed to the medication. And they typically would return back to their baseline when therapy was stopped. I hope that was what the question was addressing.
Alrighty, that seems like all the questions I see coming in at this point. Dr. Sheehan will address additional questions. If you have them, please feel free to email us at info@goldenhelix.com, and we are happy to get those answered for you. We'll go ahead and wrap up for today. I appreciate everyone's time and attendance, and we will see you next time. And thank you again, Dr. Sheehan. We really appreciate your time today.
Well, thank you so much.